This is the 16th lecture in our series on science and religion that is now in its 5th year. Before I introduce Dr. Hazen, I want to thank, as usually, our speakers, which include Hampshire College Integrated Science and Humanities Initiative, Office of the President, Office of the Dean of Faculty, the Schools of Cognitive Science, Natural Science, and Humanities and Cultural Studies. As usual, our lecture is being recorded, and we will post the video on our website, scienceandreligion.hampshire.edu, probably in a couple of weeks. The theme for this year of lectures deals with origins of life and extraterrestrials. Just a plug for our next lecture, which will be in April by Dr. Susan Clancy, and she will talk about the reasons why some people believe they were abducted by aliens. Now, for this lecture, the questions of origins are a constant theme in the dialogue between science and religion. The origin of the universe, the origin of life, and the origin of consciousness feature prominently in popular science and religion discourses. The fact that we do not as yet have specific answers to these questions also make them some of the most exciting areas of scientific research. 2 centuries ago, many considered the question of the origin of our solar system to be beyond the realm of science. Today, we have a very good understanding of the formation of our sun and the planets, and that particular origin question no longer feature prominently in science and religion discourses. Might this be true of other origin questions as well? Our speaker today, Dr. Robert Hazen, will address how scientists have been chipping away at the question of origin or origins of life. It is a great pleasure and privilege for me to introduce Dr. Hazen. And please pardon me if I take a few minutes for his introduction. Dr. Hazen is a senior research scientist at the Carnegie Institute of Washington's Geophysical Laboratory and the Clarence Robinson Professor of Earth Science at George Mason University. He received the BS and SM in geology at MIT and the PhD at Harvard University in Earth Sciences. He is the author of over 350 scientific articles and 20 books, including Genesis, The Scientific Quest for Life's Origin. So appropriate for this talk. A fellow of the American Association for the Advancement of Science, he has received the Mineralogical Society of America Award, the American Chemical Society Ipatieff Award, the ASCAP Deems Taylor Award, the Educational Press Association Award, the Elizabeth Wood Science Writing Award, and the Distinguished Public Service Medal of the Mineralogical Society of America. The past president of the Mineralogical Society of America, Hazen's recent research focuses on the role of minerals in the origins of life and co-evolution of the geo and biosphere and the development of complex systems. Normally, I would have finished the introduction, but wait. His books also include The Music Men, The Illustrated History of Brass Bands in America, 1800 to 1920, Wealth Inexhaustible, and Keepers of the Flame, The Role of Fire in American Culture, 1775 to 1925, co-written with his wife, Margaret Hinton Hazen, that explore the ties between technology and culture. At George Mason University, Dr. Hazen has developed courses and companion texts on scientific literacy. His books with co-author James Treffel include the best-selling Science Matters, Achieving Scientific Literacy, and The Sciences, an integrated approach, which a long time ago, about 12 years ago, I used it for one of my classes, but it is now in its 7th edition. He teaches courses on symmetry in art and science, on images of the scientist in popular culture, on scientific ethics, and currently teaching a class on scientific literacy. And of course, in his spare time, he has done courses for the teaching company, for which I'm an addict about that, including one on the origins of life, very appropriate for today, and on the joys of science. Okay, and just one more thing. Robert Hazen is also a professional trumpeter. He has performed with numerous ensembles, including the Metropolitan, New York City, Boston, and Washington Operas, the Royal Bolshoi, the Boston Symphony, and the National Symphony. He is presently a member of the National Philharmonic, the Washington Bach Concert, and the National Gallery Orchestra. I don't think there can be any speaker more appropriate for Hampshire than Dr. Robert Hazen. Please join me in welcoming Dr. Robert Hazen.
Thank you. Genesis. Genesis. No topic causes more conflict between science and religion than the search for life's origins and its subsequent evolution. For many people, the first chapter of the Bible is sufficient. God created the heavens and the earth. God created plants and animals. God created humans in his or her own image. But for scientists, that's not enough. We want to know how God did it. And so, when we think about the origins of life, we can imagine four possibilities. The first is that the origin of life was a miracle. A miracle is by definition something outside of natural law, outside the laws of chemistry and physics. And so therefore, by definition, it can't be studied by science. A second possibility is that the origin of life was an event that was fully in accord with the laws of chemistry and physics, and yet so rare, so improbable, that of all the billions of Earth-like planets in the cosmos, it only happened once or perhaps a few times. And if that n-member case is true, if we are alone in the cosmos, then again, it's futile for scientists to think about its origin, because how in the world can we in human lifespans reproduce a highly improbable event that required many, many chemical steps? There is, of course, a third possibility, a third n-member, and that is that life is a cosmic imperative. That throughout the cosmos, when you have Earth-like planets, when you have wet and warm planets and sufficient time, life will arise. It arises spontaneously, just like basalt arises spontaneously, and granite follows basalt. Perhaps life also is imperative. Now, if that's true, then we can imagine studying life in the laboratory. And the hypothesis I'm going to present today, because I've devoted my life to origin of life studies, and therefore I have to assume that it's something that happens commonly, frequently. It is a cosmic imperative. So we talk about emergent complexity. Life arose as a phenomenon of emergent complexity. It can be studied, therefore, in the laboratory in a sequence of experiments that isolate each of these steps of increasing complexity and therefore lead us to understanding of the origins. However, I said there are four possibilities. The fourth, as I'm sure you've heard, is intelligent design. And intelligent design is a rather different beast. It basically describes life as being so incredibly complex, having so many intricately intertwined components, that it could not possibly have arisen by any chance or random natural process. It had to be engineered, it had to be designed, and it's very much, as was described over 200 years ago, like walking along, looking down on a path of the forest floor and finding a wristwatch. And you pick it up and you look at it and you say, look at those intricate components, all the different materials that are being used to make that watch, how they fit together so perfectly, how they must have been engineered. There's a designer, and even though we don't know who the designer of this watch was, there had to be an engineer that did it. That's the argument of intelligent design. Okay, this hypothesis requires a combination of everyday physical and chemical laws, but also an unseen and unknowable designer, the intelligent designer. So the question that people have asked is intelligent design science? Now, one of the litmus tests of science is that you have to be able to falsify it. And in a curious way, intelligent design predictions are falsifiable. Someone will say, there's no possible way that the bacterial flagellum, that little long tail-like thing that lets some microbes move, could have been a spontaneous invention of nature. Therefore, it had to be designed. Now, if you can show a natural process by which the flagellum can form, then you've disproved that hypothesis. Or that an eye can evolve from a sightless being, or indeed the first cell arose. If we can show a process through laboratory experiments that shows step by step how you can go from simplicity to complexity through a purely natural process, then we can disprove those predictions. They're falsifiable. But on the other hand, intelligent design relies on that unseen designer. Since that aspect of it is really untestable, this falls outside the realm of science. And indeed, what intelligent design people will often deny, but has to be hidden in there, who designed the designers? So the implication is that someplace back in the past, there was in fact a miraculous creation of some sort. For this reason, the courts have ruled that intelligent design cannot be taught in the public classroom in the United States because there's a separation of church and state, and in a science classroom, you shouldn't have to teach religious ideas, religious doctrines. Now, this leads to the debate. Many leaders have argued that both sides should be taught. I've heard the expression, let the children decide. And they describe it as if there's evolution, there's intelligent design, these are equally weighted, and we should let people know all the different hypotheses. And in a sense, I agree with this. I think that in a philosophy course or in a comparative religion course or any course that talks about current events, it's very important for people to understand both sides of this debate. And yet scientists say things like this. Intelligent design should not be taught in high school biology classes. And what a disadvantage that places us in. It makes us look as if we're closed-minded. We're afraid of discussing alternate ideas. When in fact what we're afraid of is there's just not enough time in the school year to bring in every sort of peripheral idea, especially ones that are theologically based, not scientifically based. And yet it makes us look closed-minded. And what are we to do? What are we as scientists to do? The only answer I can come up with is that we need to design a research program that demonstrates the plausibility, in fact, the inevitability of a geochemical environment of great simplicity becoming increasingly complex through chemical reactions that are inevitable, that are deterministic, that lead from geochemistry to biochemistry. And if we can do this, if we can demonstrate step by step that life could have arisen from non-life, then there's no need for intelligent design. It doesn't disprove intelligent design. Let me give you an analogy. As you may be aware, there are theories that Stonehenge, that great Paleolithic monument in the southern plains of England, was so large and so complex it could not possibly have been built by a prehistoric civilization. Therefore, space aliens helped to build Stonehenge. The flying saucers came, they picked up the stones, set them into place. And of course, there's no way any archaeologist, anyone, can disprove this. It is a hypothesis that's out there that some people have claimed. But we can also imagine a primitive society using stone tools and rope and wood and the kinds of hard labor that 1,000 people working together can produce over many years, perhaps decades. We can imagine a procedure by which they could, using only those primitive tools, build Stonehenge. Now, we can't disprove the idea of that intelligent alien designer, but what need of it if you can show that by simple hard work and human ingenuity, Stonehenge could have been built? Why add assumptions? This is Occam's razor. This is a standard approach in science. You accept the explanation that requires the fewest number of additional assumptions, extraneous assumptions. And so we would argue, and most archaeologists would argue, that Stonehenge was just built by this human ingenuity. That's what I'm trying to do today, show you the process by which scientists are struggling step by step to learn how life could have originated from geochemical simplicity to biochemical complexity. All right, we're going to make a few assumptions today. I want to talk about emergent complexity to begin with. I'm then going to look at the emergence of simple biomolecules. We're going to look at how those molecules were assembled. That's a second emergent step. A third step is the emergence of self-replicating molecular systems. And finally, the emergence of natural selection, which follows logically from the previous one. We make a few assumptions about origin, and one of the most important ones is the idea of emergent complexity. I want to show you what is the prevailing idea about how systems can spontaneously become more patterned, more organized, more diverse, more complex. This is something we see all around us every day. You may hear arguments that this flies in the face of the second law, which says, the second law of thermodynamics, which says things become more disordered with time. But if you have a system in which energy flows through, particularly if you have a system that has many interacting particles called agents in the term of complexity, then you can actually have patterning and emergence form. We see it all the time. We see it with sand grains. Each individual sand grain only interacting with its local environment, both through gravity, through contact forces of the grain around it, through wind or waves flowing across it, and yet you get the emergent patterns of ripples and dunes, of braided streams, all different kinds of patterns you see. These are emergent patterns that arise through the flowing of energy across a lot of agents. You see it at much larger scales in terms of the spiral nebula, the armed pattern. Each individual star is only experiencing its local gravitational field and responding to that local field, and yet the collective patterning is quite amazing. And at a much more advanced level of complexity, your brain. Billions of individual neurons, each connected with others, each neuron responding to the signals it receives, and yet the collective result is consciousness. Okay, let's look at a couple of examples. My favorite is the social amoeba, Dictyostelium, a slime mold. Lives most of its life as isolated cells in amoeba-like form on the forest floor, but with various changes in local parameters, it can send out chemical signals, and the Dictyostelium cells start aggregating, coming together simply in response to their local environment, each cell only responding to the environment around it. And yet these begin to coagulate. They actually form a slug-like thing. It crawls across the forest floor. It's amazing. And then they form these beautiful stalk-like structures. New spores are released. A whole new generation of Dictyostelium is born. They melt back into the forest, go back to their single-cell form. Astonishing life cycle. That is emergence. And of course, once again, let me emphasize the human brain. You're all sitting here, and you're all an example of an emergent phenomenon because you have neurons. You have billions of them in your brain. They're connected. They're electrical impulses. There's a neural network that's formed, each neuron responding to its own region, its own part of the brain, and yet the collective is consciousness. The fact that we can sit here and think about these profound questions. We see emergent phenomena at every scale. In space, we see it in the rings and the formation of planets. We see it in the arrangements of exploding stars, and of course, in the structure of galaxies. In life, we see it at the scale of molecules, the formation of cell membranes, for example. We see it at the scale of individual cells, and we see it in the social scale of the way insects organize. I'm amazed at the behavior of ants, here tending other insects, tending aphids. Just as an aside, it used to be thought that ants, each ant communicated to other ants through chemical signals. And it turns out, it recently was discovered that ants also can emit very high-pitched sounds. So it appears that there's a kind of oral communication as well as the sensory, the chemical sense. But still, each ant is only responding to its local environment, and yet there's this collective behavior. Okay, so central assumptions of the origin of life is that the first life was based on organic chemistry, carbon chemistry. We're not going to do anything exotic with silicon or arsenic or anything else. This is basic organic chemistry 101. Life's origin relied on the raw materials available on any planet, that is water, the oceans, the atmosphere, and rocks and minerals. Nothing exotic there. And that the origin of life was a sequence of emergent steps. And the nice thing here is because each of these steps can be studied independently in a set of experiments. What are those steps? Again, let me remind you, there are 4 of them, and this is the outline of today's talk. The emergence of the basic biomolecules of life, the carbohydrates, the sugar molecules, the amino acids that build proteins, the building blocks of DNA and RNA. Then you have the emergence of organized systems. This has to do with forming polymers and membranes, the kind of structures, the macrostructures that cells need. We then have to have a system of molecules that makes copies of itself. And once that happens, the natural selection follows automatically. This is the outline of the origin of life. Now, I want to make one key point here. And this is something that's not always emphasized when people talk about the origin of life, but is coming into sharper and sharper focus. You can't have the origin of life without having many different molecules interacting, and they have to be combinatorially arranged, and then you select for things that work. And the way you make lots of different combinations and then select for things that work is to have geochemical complexities, gradients, for example, of temperature or composition. You need to have cycles, wet, dry, hot, cold, day, night. You need to have fluxes, that is flowing of fluids from one place to another. You need to have interfaces, things like crystal surfaces or water and atmosphere interfaces. You need to have surfaces where things can concentrate and organize themselves. And you need to have chemical complexity. You cannot reproduce the origin of life in a test tube sitting on a tabletop. It's a much more complex, dynamic situation. That's what geochemistry gives you. That's why the origin of life is not biology, it's geology. Okay, what we're trying to do, the first step, step number one, making the basic molecules, we need to make carbon-carbon bonds. And what you see here is a simple molecule. We start with volcanic gases, CO2, water, hydrogen disulfide, carbon monoxide, maybe ammonia, and you're trying to make, shown in green there, carbon-carbon bonds. So you go from a simple single carbon atom like CO2, then to acetic acid, maybe to pyruvic acid. We'll come back to this important 3-carbon molecule. But by making carbon-carbon bonds, you can build up all of organic chemistry. Now, the first experiments that really exploited this from the point of view of origin of life were famous experiments in the early 1950s by Stanley Miller, the young graduate student, and his Nobel Prize-winning mentor, Harold Urey. The Miller-Urey experiments are classics. I'm sure you've all heard about these. Basically, it's a tabletop piece of glassware. You have a little bit of water, which is brought to a very gentle boil. You have an atmosphere of various gases in the original experiment, ammonia, hydrogen, water vapor, methane. But other experiments have used other combinations of gases, and they all, to some extent, yield interesting organic molecules. You also have electrodes that produce little sparks, like lightning. And when you run this experiment for 2 days or 3 days or a week, you get an amazing range of organic molecules, the building blocks of life, amino acids that build proteins, sugars that do the carbohydrates, the various building blocks in the metabolic cycle, and also building blocks of DNA and RNA. They're all there. And indeed, this experiment was so successful and repeated so many times that people began to believe that this was basically the early diet. This was basically like a well-balanced meal. So it looks like the origin of life. So clearly, in the early Earth, where there was lightning, where there were oceans, where there was atmosphere, organic molecules were being made all the time. But it turns out in subsequent research, organic molecules are made in all kinds of energetic environments. In deep space, studied by Lou Amendola at NASA Ames Research Laboratory, he basically made a vacuum chamber, which is cooled down to extremely low temperatures, 10 Kelvin. He shines ultraviolet radiation on what looks like the kind of primordial dust and gas that might have been in nebulas. And he basically finds you can make all kinds of organic molecules in this environment, in this chamber. What would have occurred in vast quantities in the what are called dense molecular clouds, like the Eagle Nebula. And then you have the black smokers, the deep hydrothermal environments, which is my own personal interest, because the laboratory I come from, the Geophysical laboratory, is well known for doing high temperature, high pressure experiments. So we simulate those high pressures and high temperatures. And again, lo and behold, you can make all those different organic molecules. What we do is we start with little gold tubes. We seal ingredients, just like CO2 and water and maybe some minerals in those capsules. We heat them to high temperature and high pressure. For many years, when we started this work, Hat Yoder, my mentor and a great scientist who died much too young, at the age of 75, he still was coming into the laboratory every single day, working. He loved doing experiments, and died in a hospital of an infection that he got there, which was really tragic. But his work went on, and we did all kinds of experiments. George Cody, my colleague at the lab, also joined us in those early experiments. The three of us began Carnegie's work in astrobiology. George is an analytical geochemist who can do amazing things in terms of analyzing organic molecules. And the basic idea is we want to make carbon-carbon bonds. There are two ways to do this. One is called Fischer-Tropsch synthesis. It's used in industry. You start with a simple hydrocarbon, and you just basically build it up by adding CH2 groups and making the molecule longer like this. So that's one way of doing carbon addition. One of our very first experiments was so simple. Just put carbon dioxide and water in a tube, in a sealed tube. We also had a little bit of iron powder in that tube. Took it up to modest conditions, 300 Celsius, 500 atmospheres, ran it for 24 hours, opened the capsule and saw what we had. And this is a gas chromatogram. On the horizontal scale is time. This is a small snapshot. Every one of those peaks represents a different compound. The tall peaks, which are evenly spaced, represent long molecules. And the number gives you the number of carbon atoms. So we were making things with 20, 30, or more carbon atoms now stretched out in a long chain. And also you see lots of other bumps and wiggles here. There are alcohols, there are branched hydrocarbons, there are all sorts of interesting things. So that's Fischer-Tropsch synthesis. We know that works. Many people have reproduced this. This is very easy to make large molecules. There's a second way to make longer molecules with carbon, and that's instead of adding that CH2, you add a CO, a carboxyl group. And so here we're basically doing this hydroformylation, making larger and larger molecules, and this leads to all the carboxylic acids, which are very important in metabolism. So this is an alternate way to make larger molecules. There's a huge amount of analytical research that George Cody is the master of, but just to give you a sense of the kinds of experiments we did, this rather busy chart shows a whole series of different minerals, iron sulfide, copper sulfide, nickel sulfide, and so forth. And running these gold tubes with these various compounds, we see a competition in making longer molecules, adding carbon bonds, either through Fischer-Tropsch type addition of CH2 or hydroformylation. And the neat thing is that everything does a little bit of Fischer-Tropsch, but the cobalt and the nickel compounds are also masters of hydroformylation. You can imagine a black smoker where you're rich in cobalt and nickel and iron and copper sulfides, where these fluids are circulating around, where there are mineral surfaces everywhere, where reactions are occurring all the time, adding a CH2, adding a CO group, also adding nitrogen compounds and sulfur compounds, making more and more a rich variety of organic molecules. This happens so easily. It's also interesting for those of you who are into chemistry that the reason cobalt and nickel work is because their particular electron configurations are particularly attract the CO groups, and so they're very, very good at hydroformylation relative to the other transition elements here. So there's a rich chemistry going on here, and we conclude just from every one of these experiments, and many more done around the world, that prebiotic chemistry, making these molecules, is easy. That problem is solved. You can make amino acids, you can make sugars, you can make bases, you can make the basic carboxylic acids. All of these compounds that we need for the origin of life were present in the prebiotic earth. And I'm delighted as a mineralogist to say that minerals did play a critical role here. And by the way, you will see in the origin of life research a phenomenon that I think is delightful, tells a lot about human nature. Nobody gets their PhD in origin of life. You get your PhD in chemistry or in biology or in my case mineralogy or something else. And every one of us seems to see the origin of life in terms of whatever PhD we got. I know people who are done molecular biology where they study DNA and RNA. For them, the RNA world is the only way to think of the origin of life. There are chemists who specialize in lipids, and for them it's the lipid world. I'm a mineralogist. Minerals are the key to the origin of life. I'm sorry, it's true. It has to be true, but it's fun. We sort of smile when we say these things, but we do have that predisposition. Okay, so step 2. This is the emergence of organized molecular systems. And here we have a problem. Making the molecules is easy. The trouble is you make too much. Instead of making the few hundred molecules that are found in living things, an E. coli, that classic microbe, has only 500 different kinds of small molecules up to about 12 different carbon atoms. Now, that's in contrast to the catalog of organic molecules, Beilstein, which lists over 6 million organic compounds. Life is incredibly picky, it's finicky, it's idiosyncratic. So our problem here is not making the molecules, it's we make too many different kinds. So how in the world do you get the selection of just a few molecules out of that prebiotic soup that had probably hundreds of thousands or millions of different species? What process could have done that? Okay, one of the possibilities is self-organization, self-selection. And let me tell you a little story about this. One of the very earliest experiments that George Cody, Hat Yoder, and I did was with pyruvic acid. Remember, that's that 3-carbon molecule. It's very important in metabolism. And what we said, what we'd really like to do is take the 3-carbon pyruvic acid, put it in a capsule with lots of CO2, and try to make the 4-carbon molecule oxaloacetic acid. That turns out to be a key step from 3 carbons to 4 carbons in what's called the reverse citric acid cycle, which you'll see later on in this talk as one of the ideas that's central to some ideas of origin of life, that was the first metabolic cycle. You have to be able to keep adding carbon atoms from pyruvic to oxaloacetic. And we thought, this is great. We're going to just run this reaction. We're going to do it at 200 Celsius, 2,000 atmospheres, 2 hours, very simple conditions. And if we convert pyruvic acid to oxaloacetic acid, we'll get a paper in Science or Nature, headlines, problem solved. That's not what happened. Ran those capsules for 2 hours, and when they came out, they were all puffed up. They were just like they were ready to burst. So we knew there was a lot of internal pressure. And I said, uh-oh, this is going to have to be careful. So you put it in liquid nitrogen, you cool it down, and even after you freeze it, you take a pair of clippers and you snip off the end. Bang! There's all this pressure, and then you drop the capsule into the bottom of a vial and just let it sit there. And after about 30 seconds, as it thaws, it starts to and all this yellow-brown oily goo starts pouring out of the capsule. I mean, you couldn't believe it. Just a few drops of pyruvic acid we begin with, and it just looked like it just kept coming and kept coming out like this. And so you can see this smear on the bottom. And then the aroma. The aroma, it is so, at lower temperature, like 200 Celsius, it smells like molasses. It's very sugary, it's very sweet. At about 300 Celsius, though, it smells exactly like Jack Daniels. And it's so cool because people, and it just permeates the lab. People would come by and they'd sniff and they'd say, "Can I have some?" And it's just so fascinating. Well, what you're doing is you're making a lot of really interesting organic molecules. You're not making oxaloacetic acid. You're doing something else. And in fact, you're making an embarrassment of riches. Here's the GC trace. And there are thousands upon thousands of different compounds here. And this is because the pyruvic acid polymerizes and it cyclizes and it does other sort of split apart and reforming in various ways. And so you can analyze these individual molecules and it's just a mess. And the trouble is, as I said, life doesn't want a mess. It wants just a few things. So how in the world could this be of any use? And I sort of shook my head and I said, wow, this is discouraging. And so I guess we're going to go nowhere. And I told my good friend David Deamer at UC Santa Cruz about this. Dave Deamer is a person who's an expert on self-organization of what are called lipid molecules. And he said, well, have you put the stuff in water? I said, what do you mean, Dave? He said, well, maybe it'll self-organize. So I did this. I also worked with my friend Marilyn Fogel, who helped us analyze what we had. And it turns out that in this mess of stuff, we had a lot of long-chain molecules that had 8, 10, 12 carbon atoms in a row. You notice they're both smiling? People in the origin of life field are always smiling. It's so much fun. It's so interesting. Sure, I mean, this is a fundamental, profound question, but if you're not having fun, you're doing something wrong, and my friends, we smile a lot. Anyway, so he said, Dave Deamer said this is what's going to happen. You put these molecules in water, and maybe some of them will form little vesicles, little cell-like enclosures, and that's a key step in the origin of life. Even though you have a mess, maybe that mess will self-organize, and lo and behold, this is what happened when we put that stuff into water. It self-organizes. They're cell-sized structures, and also they fluoresce, which they're just beautiful under the microscope. So these aren't cells, by the way. I mean, this is just a sphere of these molecules that have selected themselves, found them, and organized. But we see this all the time. We see this in the kinds of lipids that come from meteorites when they're extracted. We see it in our experiments. We saw it in Stanley Miller's experiments. This is a deterministic part of the natural world. Making these self-organized structures, which can serve as the precursors to a membrane for a cell, is an automatic part of our natural world. Okay, so that's one step. The second possibility, though, is that there are many different kinds of molecules that are water soluble. They're not going to form self-organized spheres or something of that sort. So the amino acids, the sugars, these are things that are critical, and yet how in the world do you select them out of a very dilute prebiotic soup? So one of the hypotheses I propose that perhaps minerals can help us in this, because the ancient environment was the floor of the ocean just covered, of course, with minerals of all different kinds. And they had lots of interesting surfaces. How could this work? Well, so here's the biggest challenge in molecular selection. It's not just how do you get the right amino acids. It's how do you get the right handedness of amino acids, because biological molecules often come in mirror-related pairs, right-handed and left-handed molecules. And for some reason, life only uses the left-handed amino acids, and it almost always uses the right-handed sugars, and not the mirror-related pair. So you have to figure out, when did that happen? Could there have been a prebiotic mechanism to select right and left? Well, here's what different molecules look like. You have a central carbon atom and 4 things arranged clockwise or counterclockwise. So the big question we're asking is, how did life on Earth become homochiral is the word. It became handed. But it turns out our work has been followed very, very closely by the pharmaceutical industry, because we as a nation pay $200 billion a year for chirally pure pharmaceuticals. Levothyrox, left-handed thyroxine, right-handed molecules as well. And the reason is because chirality is important. This is a very classic and fun example. Limonene, the right-handed form, smells like oranges and is used as a flavor or fragrance in that way. The left-handed version smells like lemons, because the receptors in our nose are handed and the molecules are handed, and so it fits in like a glove. You know, you can't use your right hand with a left-handed glove, at least not very easily. There's a more sinister example, thalidomide, which you've heard about, caused birth defects. The right-handed form cured morning sickness. Trouble is, even if you had a pure right-handed form, in your body it switches from right to left in about 24-hour half-life, and the left-handed form was the one that caused these birth defects. So chiral purity is really important. Now, here's the problem. Prebiotic syntheses, every one that I've told you about, Miller-Urey or hydrothermal or deep space, forms equal amounts of left and right-handed molecules. But life is highly selective, using only left-handed amino acids, right-handed sugars. So what's the mechanism of what we call symmetry breaking? And I propose that one possibility, one of many possibilities, is mineral surfaces. Now we know that, for example, the commonest mineral of beach sand, quartz, has left-handed crystals and right-handed crystals, because the structural elements has a helix in it, and the helix can either spiral to the left or spiral to the right. That means every surface of quartz can select chiral molecules. Now here's an example of why this is so important. I've been a mineral collector for a long time, and when I was a teenager I collected this sample of quartz in Paterson, New Jersey. If you look carefully at those little quartz crystals about 1 millimeter across, you see that there are 6 triangular faces coming to a point. In this case, 3 of them are coated with iron oxide and 3 of them are not. That's an important message, because it means that each face of the crystal, this by the way has 3-fold symmetry, not 6-fold symmetry, the quartz crystal, each face has a different structure and therefore is going to select molecules differently. What everyone used to do was to take crystals of quartz, crush them to the finest powder they could, and then run an experiment. Now that gave you a lot of surface area, which is good in an experiment like this, but it destroyed all that selective information about which face was doing what. So we've been doing experiments on large single crystals where we have individual faces that we can study. So we look at, for example, Feldspar, the commonest mineral, and the M faces here are mirror symmetry faces. We've been looking at pyroxenes, the commonest minerals of magnesium and iron. Again, the M faces are mirror symmetry faces. And I've been looking particularly at calcite, calcium carbonate. This is the mineral of snail shells and clam shells and so forth, coral reefs. And it has, in its commonest form, lots of chiral faces. Those faces have a very strong ability to select and concentrate left-handed molecules. So an experiment that we did some years ago was taking a large crystal of calcite, about fist-sized crystal, and it had 12 faces on it, 6 here, 6 here. We took that crystal and put it into a beaker, which had a 50-50 mixture of left-handed and right-handed amino acids. Let it sit overnight, came back the next day, pulled it out, just rinsed it off, and then very carefully, face by face, put a few drops of hydrochloric acid, let them sit there, pulled the hydrochloric acid off. That strips off the outer layers of the calcite and also pulls any amino acids that are bound to that layer. Put it in a vial, and step by step, face by face, going around the crystal, looking at each of those faces. And what we found, is a complicated experiment, but the results are simple, is that we have left-handed and right-handed amino acids absorbing differentially on those different faces. Now, on the early Earth, there are equal numbers of left and right-handed faces. But what you're doing is you're creating local environments, microenvironments, where you have experiments going on. Experiments in concentrating left-handed amino acids, experiments in concentrating right-handed amino acids. And one of those experiments, we think, probably took off. It learned how to juxtapose amino acids in just the right way that you could go on to the next step. So, by the way, we can also analyze this. This is a calculations using density functional theory to understand exactly how the molecules are sticking to the surface. So what you have below here are a representation of the calcite surface, and the molecule, in this case aspartic acid coming in. D means the right-handed version, L is the left-handed version. Turns out in this particular configuration, the D version, the right-handed version, has 3 strong points of interaction. The left-handed version, because of its shape, only has 2. And the result is a difference in surface energy of 8 kilocalories per mole. In human terms, that's a lot. It means that at room temperature, you're going to have a 99% selection on one face versus the other. Really quite an extraordinary effect, and one that I'll tell you again, the pharmaceutical companies are very interested in. Okay, so the conclusion of this part 2. We don't know all the details, but it's clear that you can select and concentrate molecules either by self-organization or by adsorption onto a mineral surface, or perhaps absorption onto a water atmosphere interface, all different sorts of ways to do this. So in principle, this is a solved problem. The details, of course, are fascinating, and we continue to work on that. Now, the next step, and I'll tell you right up front, this is one that has not been accomplished yet. A lot of people are working on it. It's a fascinating one, is making a self-replicating system. Because after all, that is the characteristic of life we're most interested in. A group of molecules that can make copies of itself, more and more copies, and therefore take over as sort of the first, at least primitive, life form. Many people think that this reverse citric acid cycle, or TCA cycle, is the core. But the holy grail is to get one of these systems working in the laboratory. This will make big headlines in all the newspapers when it happens. I'm greatly influenced by Harold Morowitz, a theoretical biologist at George Mason University, who has championed this idea that the reverse citric acid cycle is the core metabolism of all life. It's in all of our cells. It's used by many microorganisms today. It seems like a biochemical fossil. And if you can get the TCA cycle to work in some environment, then all other biosynthesis radiates out from it. You make your amino acids, then the amino acids can be formed. You can make lipids. You can make any of the building blocks that you need for life from this core metabolism. So this is a fascinating idea, but it's not the only one. By the way, here's some experiments. This incredibly busy chart that George Cody made shows some of the reactions that we've gotten to go. Embedded in this graph are most of the steps in that reverse TCA cycle. In fact, there are only 2 that we haven't gotten to go yet. One of them is that important up at the top. If you can see, there's pyruvic acid going to oxaloacetic acid. Still haven't been able to get that one to go. And so this is a continuing area of research. Perhaps there's another even simpler cycle nested in here that we can find someday. But this shows you the kind of research, step by step, look for these reactions, see if we can get this to go in a plausible geochemical environment. Here's another idea. Stuart Kauffman at the Santa Fe Institute proposed that instead of a cycle of just a few molecules, perhaps there was something he called an autocatalytic network. And the idea here is you take advantage of the fact that the prebiotic soup has tens or hundreds of thousands of different molecules. And he said if some subset of those molecules, perhaps 1,000, perhaps 10,000 of them, were autocatalytic, that is, molecule A helps synthesize molecule B, B helps synthesize C, C helps synthesize D, and you get a network where they all eventually connect up to each other. So this is just a tiny part of one of these networks. That you'd have a system in which certain molecules benefit and get larger and self-replicate in the sense that they're catalyzed by the molecules in the network, and any molecule not in the network degrades, goes away. And then a network like this will evolve simply because you find networks within the network, and it gets smaller and smaller, and maybe perhaps eventually reduces down to that reverse citric acid cycle. That's a hypothesis, but it's virtually impossible to test in the laboratory, because how in the world do you put thousands of molecules into your analytical apparatus and then see how they're all changing with time? It's a very difficult concept, but it's possible because the early geochemical environment was complex. And then there's the RNA world. And many people advocate the very plausible idea that the very first self-replicating molecule was something like RNA. The reason is because RNA is an amazing molecule that both carries genetic information, but also can catalyze reactions in the cell. And if you could find an RNA molecule that can catalyze its own reproduction, then you have something that really looks like a living system. It has genetic information and it makes copies of itself. So a single RNA molecule, perhaps encompassed in a membrane, could be a plausible first life form. So many people promote the RNA world scenario. It's probably the idea to beat in the origin of life field. The one problem is that there's no known way right now to make RNA in a plausible prebiotic environment. But a lot of people are working on this, and I think over the next decade we'll have a much better idea if some of these ideas come to fruition. If someone can show, yes, RNA can be made easily under a certain set of circumstances, then that will be pretty much accepted. But it's still, this is a big challenge. It's a big hurdle right now. And you have to ask, therefore, we know what happened. We have a prebiotic soup. We see some self-organization going on. And then you have a first self-replicating system. What happened in between? That's a big mystery. So the conclusion of step 3 is we have some good ideas, but we haven't done this yet. I think we may be getting close. I would not be surprised if in a decade or so, in the next decade, this is solved or the first self-replicating system is shown in a laboratory experiment. If so, as I say, it'll get a lot of headlines. But then step 4. To me, this is a no-brainer. Once you have a self-replicating system of molecules, it's going to evolve. It's going to evolve because simple organic molecules have all sorts of variants. You can stick a sulfur for oxygen. You can add a CH2 group. You can make it a little bit different. Mutations are going to occur. And as a result, those mutations then let competition and selection run rampant in a system. And you say, well, that's just arm waving. You know, you can say that, but how do you know? Well, in fact, it's been done. It's been done in the laboratory, and it now makes people a lot of money. This is Jack Szostak, one of the real heroes of the origin of life community. He also won the Nobel Prize a couple of years ago for other work that he's done, so he's a very brilliant and interesting guy. And he demonstrated molecular evolution through what is called Aptamer evolution. Now, Aptamer is a molecule that has a specific function that does something. In this particular experiment, what the molecule does is it recognizes and latches on to a different molecule. So you may have heard of the molecule ATP. It's used in energy production in cells. He basically had a variant called something GTP, and it's a molecule he sticks to the side of a tube. And then he has long strands of RNA, 100 RNA letters long. This is a long polymer, and these things automatically fold up into various complex shapes. Some of them, a very, very tiny fraction of the molecules, fold up in a shape that latches on to that GTP just perfectly. Most of them don't. And the thing you should know about RNA is there are 4 different RNA letters, and those 4 different letters can occur in each of the 100 different places. So there's 4 to the 100th power different possibilities of RNA strands. Now, if you were asked to design an RNA strand that exactly latched onto a GTP molecule, you could use all the computer power in the world for 1,000 years, and we still wouldn't be able to do it. There's no known way to determine this. So how would you do this? You use molecular evolution. You start with a random pool of 10 to the 14th different randomly generated strands that are 100 RNA letters long. And they fold in all different ways. And you have no idea which ones of those strands might work. But if you expose the molecules, this is done with little beads that have been decorated with the GTP molecules. So the GTP molecules are sticking out. Think of them like hands sticking out. You're trying to find a glove that fits neatly onto the... Okay, and what you find is that a few molecules out of that 10 to the 14th happen to stick to the GTP. You wash the beads, and only the RNA molecules that have folded up and sticking to the GTP stay on. And then you basically recover those beads through standard molecular processes. You recover those molecules. You then do various genetic tricks to amplify the number of molecules with mutations. And you create another population of 10 to the 14th RNA molecules. But this time, every RNA molecule is a mutated variant of one that actually had stuck before. Get what I'm saying? So you've now made a new population, but now all of these have the potential to stick. And you find that in the second cycle, a few of those stick much better. And if you do this only a half a dozen times, out of the 4 to the 100th power different combinations, you will zero in on RNA molecules that bond with the absolute maximum possible energy to that. It's amazing. I mean, it takes, hours, a few hours to do this sort of thing. Well, a few days, but it's now used as a medical technique to target specific viruses. So it's worth a lot of money. And Jack Szostak and others have certainly benefited from this, but it's an amazing thing. So this is molecular evolution. The exact same kind of thing would have happened on primitive Earth. Now, one of the things I want to come back to is, remember I said geochemical complexity is really important in origins because you have to have a selective process. Well, think about that Aptamer evolution work in terms of gradient cycles, fluxes, interfaces. First of all, you start with chemical complexity, that random pool of 10 to the 14th. You need to have a large number of different combinatorial possibilities to select from. You also have interfaces or surfaces. That's the surface of the glass bead where you can connect to the different molecules. You have fluxes, that is washing the sample over and over again. You have cycles of 2 different kinds here. You see, this is essential. You can't have evolution without this kind of geochemical complexity layered on top of it. Okay, so life's origins, the 4 steps. Synthesis of biomolecules. That's easy. We know how that's done. Second step, biomolecular selection. Well, we know 2 different ways, self-selection, and we also have mineral surfaces or other kinds of surfaces. A lot we still need to learn, but the basic principles are well understood. Then the big uncertainty, and that's how you get from there to a self-replicating molecular system. And finally, once you have a self-replicating system, molecular natural selection follows automatically. So this is our outline of the origin of life. And I think I've shown you experimentally how this may happen. But an exciting development, this is not the way I was normally going to end this talk, but there's an exciting development that has recently come up, which I think may short-circuit some of this work. And this is the result of a new initiative that I'm involved in called the Deep Carbon Observatory, sponsored by the Alfred P. Sloan Foundation. It's a big international effort to understand carbon, including trying to understand deep life. So we have a whole bunch of people who are doing things like drilling and bringing up microbes from deep drill holes. And what they find is if you have a drill hole that goes down deep to where the temperatures are up to 130, 140, maybe even 150 Celsius, you find viable microbes. It's amazing. There's an incredible domain of microbial life deep underground, several miles down in some places. If you go deeper, you go above 150 Celsius, there no longer appear to be cells. But there is some indication, it's very hard to do these analyses, but there's some indication that there's something lifelike going on. There are things that look like polymers or biofilms or membranes or something down there. The analytical tests are not very well defined. There's always the possibility of contamination. These are being retested. But in order to test this, there was an experiment that's now being run on the Juan de Fuca Ridge. This is one of these black smoker places where there was this chimney. They drilled a hole in the side of the chimney, so the outside temperature is about 4 Celsius. The inside temperature is close to 300, and there's this long experimental apparatus that has separate compartments. Each one is going to be at a higher temperature. Little chips in the mineral chalcopyrite, a sulfide which microbes can grow on. You stick that into the tube, and the water is flowing from the high temperature end out to the cold temperature end. And after 6 months, in the first set of experiments where they pulled this out, at the low temperature, up to 50 Celsius, they found microbial biofilms. From 50 to 100, microbial biofilms. From 100 to 150, again, you find microbes living and forming biofilms. At the higher temperature, you don't see organized cell structures, but you still find biofilms. Something is happening. There's some kind of molecular organization occurring at much higher temperature. Well, we don't know. It's possible. It is possible that this represents a kind of self-replicating or self-organizing system. I don't know whether you want to call it life or proto-life or something like that. But is it not possible? Cellular life could really be limited to 130 or 140 Celsius simply because of all the complex chemical machinery inside the cell. But is it not possible that there are simpler forms of self-replicating, self-organizing molecules at higher temperatures, maybe 150, 200, 250 Celsius? And if that's true, there have been continuous domains in Earth since the origin of life, where there are domains that are at those higher temperatures. They're physically continuous, and so they would have persisted, and they'd be beyond the predation of cellular life. So they should still be there today. So this search for this much more primitive, possibly self-replicating organic systems, things that are forming at these higher temperatures, is certainly one of the driving forces behind our Deep Life initiative. And just to give you a sense of what this might mean, cellular life could conceivably exist as deep as 40 kilometers in a very rapid subduction zone where the temperatures would be 130 to 140 Celsius. But if we could have life existing in this more primitive form, you could have perhaps a life-like structure as deep as 200 kilometers in Earth's mantle. This is an astonishing idea. It's something we really want to explore. I think it opens the door to thinking about origin of life and life on Earth today in ways that we just haven't thought of before. It's a very exciting frontier. So if you want more information about the Deep Carbon Observatory, please go to this website, http://dco.ciw.edu. My conclusions, I think they're pretty straightforward. The origin of life is best understood as a sequence of emergent events going from simple to complex. We see these stages, each of which can be studied experimentally. We don't have all the details yet. But of course, what would be the fun of science if we knew all the answers? So the neat thing about getting up in the morning and going into the laboratory is searching those things we don't know. Finally, I want to end on a little bit more philosophical note. I know that this idea of the origin of life being a purely chemical, physical process is disturbing to some people. How can we be created in God's image if this is a mere chemical process with some random component? Molecules here and molecules there just happening to juxtapose in a way that leads to something that self-replicates and maybe then goes through an evolutionary process. How can we be created in God's image? What can be the meaning of life if it's this random, undirected process? Well, I want you to think about the origin of life in a different way, in a deterministic way. We live in a universe that began with the Big Bang and the formation of vast amounts of hydrogen. Those hydrogen atoms inevitably, inexorably formed giant stars. Those giant stars inevitably, inexorably went through a process of nucleosynthesis in which the entire periodic table of the elements was formed, and those stars exploded and released that richness into the cosmos. And then solar systems followed. Earth-like planets inevitably, inexorably formed in the cosmos. At current estimates, there could be 1 billion trillion Earth-like planets in the visible universe. And so for Earth-like planets, life arises inevitably. And from life, evolution, and from evolution, consciousness. And so we are living in a universe that is learning to know itself. Thank you very much.